it's me, Erica Moore, and welcome back to my channel. So, I am back again for another discussion, and for this video, we are going to talk about the cellular respiration and the fermentation.
Every task performed by living organisms requires energy. Energy is needed to perform heavy labor and exercise, but humans also use energy while thinking and even during sleep. In fact, the living cells of every organism constantly use energy. Nutrients and other molecules are imported into the cell, metabolized or broken down, and possibly synthesized into new molecules, modified if needed, transported around the cell, and possibly distributed to the entire organism. In the process of photosynthesis, plants and other photosynthetic producers create glucose, which stores energy in its chemical bonds. But once photosynthesis has created glucose to store energy, both plants and consumers such as animals undergo a series of metabolic pathways, collectively called cellular respiration, to use that energy. Cellular respiration extracts the energy from the bonds in glucose and converts it into a form that all living things can use. Cellular respiration is the process by which organisms combine oxygen with foodstuff molecules, diverting the chemical energy in these substances into life-sustaining activities and discarding, as waste products, carbon dioxide, and water. Organisms that do not depend on oxygen degrade foodstuffs in a process called fermentation. One objective of the degradation of foodstuffs is to convert the energy contained in chemical bonds into the energy-rich compound adenosine triphosphate, or ATP, which captures the chemical energy obtained from the breakdown of food molecules and releases it to fuel other cellular processes. In eukaryotic cells, or any cells or organisms that possess a clearly defined nucleus and membrane-bound organelles, the enzymes that catalyze the individual steps involved in respiration and energy conservation are located in highly organized, rod-shaped compartments called mitochondria. At the heart of ATP is a molecule of adenosine monophosphate, or AMP, which is composed of an adenine molecule bonded to both a ribose molecule and a single phosphate group. Ribose is a 5-carbon sugar found in RNA, and AMP is one of the nucleotides in RNA. The addition of a second phosphate group to these core molecules results in adenosine diphosphate, or ADP. The addition of a third phosphate group forms adenosine triphosphate, or ATP. Biologists differ somewhat with respect to the names, descriptions, and the number of stages of cellular respiration. The overall process, however, can be distilled into 3 main metabolic stages or steps. Nearly all of the energy used by living things comes to them in the bonds of the sugar glucose. Glycolysis is the first step in the breakdown of glucose to extract energy for cell metabolism. Many living organisms carry out glycolysis as part of their metabolism. Glycolysis takes place in the cytoplasm of most prokaryotic and all eukaryotic cells. Glycolysis, which is also known as the glycolytic pathway, is a sequence of 10 chemical reactions taking place in most cells that breaks down a glucose molecule into 2 pyruvate or pyruvic acid molecules. Energy released during the breakdown of glucose and other organic fuels molecules from carbohydrates, fats, and proteins during glycolysis is captured and stored in ATP. In addition, the compound nicotinamide adenine dinucleotide is converted to NADH during this step. Pyruvate molecules produced during glycolysis then enter the mitochondria, where they are each converted into a compound known as acetyl coenzyme A, which then enters the TCA cycle.
Next is the tricarboxylic acid cycle. The TCA cycle, which is also known as the Krebs or citric acid cycle, plays a central role in the breakdown or catabolism of organic fuel molecules. The cycle is made up of 8 steps catalyzed by 8 different enzymes that produce energy at several different stages. Most of the energy obtained from the TCA cycle, however, is captured by the compounds NAD+, and flavin adenine dinucleotide or FAD, and converted later to ATP. The products of a single turn of the TCA cycle consists of 3 NAD+ molecules, which are reduced through the process of adding hydrogen or H+, to the same number of NADH molecules and one FAD molecule, which is similarly reduced to a single FADH molecule. These molecules go on to fuel the third stage of cellular respiration, whereas carbon dioxide, which is also produced by the TCA cycle, is released as a waste product. And lastly, the oxidative phosphorylation. In the oxidative phosphorylation stage, each pair of hydrogen atoms removed from NADH and FADH provides a pair of electrons that, through the action of a series of iron-containing hemoproteins, the cytochromes eventually reduces one atom of oxygen to form water. In 1951, it was discovered that the transfer of one pair of electrons to oxygen results in the formation of 3 molecules of ATP. Oxidative phosphorylation is the major mechanism by which the large amounts of energy in foodstuffs are conserved and made available to the cell. The series of steps by which electrons flow to oxygen permits a gradual lowering of the energy of the electrons. This part of the oxidative phosphorylation stage is sometimes called the electron transport chain. Some descriptions of cellular respiration that focus on the importance of electron transport chain have changed the name of the oxidative phosphorylation stage to the electron transport chain. Some living systems use an organic molecule as the final electron acceptor. Processes that use an organic molecule to regenerate NAD+ from NADH are collectively referred to as fermentation. In contrast, some living systems use an inorganic molecule as a final electron acceptor. Both methods are called anaerobic cellular respiration, in which organisms convert energy for their use in the absence of oxygen. Lactic acid fermentation. The fermentation method used by animals and certain bacteria, like those in yogurt, is lactic acid fermentation. This type of fermentation is used routinely in mammalian red blood cells and in skeletal muscle that has an insufficient oxygen supply to allow aerobic respiration to continue. In muscles, lactic acid accumulation must be removed by the blood circulation and the lactate brought to the liver for further metabolism. This is the chemical reactions of the lactic acid fermentation. The enzyme used in this reaction is lactate dehydrogenase, or LDH. The reaction can proceed in either direction, but the reaction from left to right is inhibited by acidic conditions. Such lactic acid accumulation was once believed to cause muscle stiffness, fatigue, and soreness. Although more recent research disputes this hypothesis. Once the lactic acid has been removed from the muscle and circulated to the liver, it can be reconverted into pyruvic acid and further catabolized for energy. Lactic acid fermentation is common in muscle cells that have run out of oxygen. Alcohol fermentation. Another familiar fermentation process is alcohol fermentation that produces ethanol. Because of this,
This kind of fermentation is also sometimes known as ethanol fermentation, and there are two main reactions in alcohol fermentation. The first reaction is catalyzed by pyruvate decarboxylase, a cytoplasmic enzyme with the coenzyme of thiamine pyrophosphate, or TPP, which is derived from vitamin B1 and also called thiamine. A carboxyl group is removed from pyruvic acid, releasing carbon dioxide as a gas. The loss of carbon dioxide reduces the size of the molecule by 1 carbon, making acetaldehyde. The second reaction is catalyzed by alcohol dehydrogenase to oxidize NADH to NAD+, and reduce acetaldehyde to ethanol. The fermentation of pyruvic acid by yeast produces the ethanol found in alcoholic beverages. Ethanol tolerance of yeast is variable, ranging from about 5% to 21%, depending on the yeast strain and environmental conditions. This is the diagram of alcohol fermentation.
So that's all for this video. I hope you learned a lot about our discussion today. And see you in my next vlog. Thank you for watching. Bye.